Good afternoon to you all. I hope everybody is well and safe. And welcome to our biology revision session with me, Danian Singh, and Miss Zafia Naz. Right, we both belong to the Department of Sciences, and we teach various levels or various streams within biology. For example, I teach ecology and conservation, whereas Ms. Zafia teaches genetics, physiology, and also conservation. So we'd like to welcome you all, and most probably some of your friends might be joining in a bit later. Okay, but we'll make a start. It's already 3:30. So I hope everybody is well and you're all able to hear and see me. Okay, if there is any network issues, you're not able to see me, or if you have any issues, please, you can either type it out in the chat option, right, that is available, or you can always turn on your mic to let me know if there are any issues, me or Miss Zafia. Okay, so we will begin, right? Well, you have your exams next week, so I hope that you've been studying up and you have been making short notes, right? You're going over the biological concepts. Okay, I know that biology is a lot of theory, right? A lot of theory. And sometimes what happens is that you get lost within that theoretical bit here and there. So it is always advisable, right? And this is what I tell my students as well, that it is advisable to have a small notebook, right? And in that, you can make your short notes. You can have your hand-drawn diagrams, especially for plant life cycle, right? I know that you have an angiosperm and gymnosperm life cycle, which is a bit complicated, okay? But if you hand draw it, if you draw it with a flowchart mechanism, then it will be something easier that you can actually understand, okay? So what me and Ms. Zafia have done is that we have divided the topics amongst us, and we're actually going to discuss various components based on the year 13 biology curriculum. Okay, so is that okay with everybody? Anybody has any questions for now before we actually start the discussions? Anybody has any questions? Okay, the recording will be available to you tomorrow through the University of Fiji website, just in case if you have missed certain components of this discussion session or revision session. The recordings will be available to you via the university website, so you can access whichever subject that you missed out on, or if the recordings, if you want to download and keep it, right, the recordings will be available to you via the website. Okay. So I think there are no questions, so I'll make a start, and then Ms. Zafia will join in a bit later. Okay, so I'll share my screen with you guys, and if you have any questions, please make sure you can type it out in the chat option. I'll go back and forth from the chat option to my presentation so that I know if you have any questions, I'll answer the questions simultaneously, as well as discuss the various concepts with you with that. Remember, during the discussions, this is sort of like an open discussion. Remember, you can turn on your mic and ask me questions if you have any, right? Or if you do not want to, then you can use the chat option that is available. Okay, I will also ask you some questions during the session. Okay, so make sure that you at least have some idea about these concepts, okay? Because you've gone over these concepts as well. But if you do not know, please, you can ask me to repeat, right? If you have any clarifications, if you do not understand any terms, please do ask me to repeat, or you can ask me to clarify, right? During my discussion, you can interrupt me whenever you want to, okay? There is no hard and fast rule, and you can always ask me questions or any issues that you have regarding the topics that we're actually going to discuss, okay? So just give me a moment, give me a second. I'll just present the topics to you, the topics that I will be discussing. Okay. So my screen should be visible to you now. Okay, so I'll just share my presentation with you, right? So if you have any questions, any issues, feel free to ask me. Like I said, right, you can interrupt me whenever you want, right? This is a very open session. And at the end of the day, it's about you learning these concepts and the ability to master these concepts. So this is the biology revision class, right? And the topics that I'll be discussing with you is mostly natural selection, evolution. I'll be also going over viruses, biomes, and very briefly we'll discuss the plant kingdom. Okay, so these are some of the topics. I've asked a couple of year 13 students, and these are some of the topics that they find difficult, or these are the topics that they actually have some issues with. So we'll go over these topics, and I will ask you some questions regarding these topics, and make sure that you can try to answer them. If you don't know, it's still fine. We'll discuss it. We'll find out the difficulties that you have in these topics or in these issues. Okay, so we'll move from natural selection, right, and we'll go down to the plant kingdom, right, and the remainder of the topics, the remainder of the important topics will be discussed by Ms. Zafia. Okay, so we'll start off with natural selection. Okay, natural selection is probably one of the most important topics in biology, right? Whether you are in high school, whether you are in university, okay, natural selection is a topic that it's going to be discussed over and over again. Okay, and the definition as well as maybe an example of natural selection is important for you to know, so you can answer the questions. Okay, so whenever a question states natural selection, okay, whenever a question states natural selection, make sure you know what it actually means, right? Natural selection, natural means nature. Okay, natural selection means nature. Okay, it is where nature selects the organism that is the most fittest. Okay, remember, nature selects. The opposite to natural selection is artificial selection or selective breeding. Okay, there is a difference between the two, right? They are opposite to each other. Natural selection, remember, is where nature selects the organism that is the most fittest. Whereas artificial selection is where humans select features that are beneficial to them. Okay, artificial selection, for example, selecting the various types of vegetables, right? Most of the vegetables that you eat on a daily basis, remember, they have been artificially selected for you by farmers. Okay, whereas natural selection, nature selects, right? Now, this is more of a very brutal or a very, you can say, evil side or a very bad side of nature, because nature will totally remove organisms that are not able to adapt to the environment. Okay, and some species or some traits, for example, will become very dominant. Okay, will become very, very dominant in nature. Okay, and eventually, right, those traits or those species will then take over. Whereas the ones that are not able to adapt, remember, they will not be able to survive. So I've given you a very simple diagrammatic form so that it's easy for you to actually understand, right? So let's say, right, there is a rat population. Okay, so what do you mean by population? Can you tell me through the chat maybe, what do you mean by population? Okay, what do you mean by population? Okay, population is a group, isn't it? But what is the definition for population? Is it different? Okay, somebody has mentioned group of people. Okay, yes. Okay, so based on that, okay, somebody said group of organism. Okay, yes. Is it a group of the same species or is it different species? Let's say either a group of minor birds or let's say a group of organisms living in the forest. Okay, yes, somebody has mentioned a correct answer, right? Same, right? A population, remember, for example, population of humans. Somebody said group of humans or group of people. right? So a population of humans, right, of the same species. Okay, remember, the same species, right? It does not involve two different species or different species. Okay, if you have a group of minor birds and a group of red-vented bulbuls, for example, they do not form a population. A group of minor will form a population. A group of red-vented bulbul will form a population. Like two separate organisms, right? They'll form their population. So we'll get back to the presentation. Okay. So you have a group or a population of rats, let's say, living in an area. The background of the area is black or grayish in color. Okay, you can see there are two different variations, or you can say two different fur color. One group or one type of a rat here has a sort of like a purplish or a peach color fur color, whereas the other has a grayish, you can say, or a blackish fur color. But remember, they belong to the same species. The only difference is that the fur color is different. Okay, only the fur color. The difference is the fur color. So this peachish or this brownish, light brownish fur color, remember, is a type of trait. Whereas this black fur color, remember, is a type of trait as well. Okay, so there is a variation within the population. Okay, there is a variation within the population. So what happens is that the background is black in color. Okay, the background is black in color. So what actually happens is that there is a predator. Okay, there is a predator. That predator will be able to spot, right, which fur color the most. It'll be able to spot the peach color first. Okay, because it is quite visible. Okay, it is very conspicuous. Okay, you can actually see it from far. So what will happen is that that bird will prey upon this colored rat first. Okay, because it is easy to spot and easy to catch. Okay, and what will happen is that after some time, the individuals that have this brownish fur color will eventually decrease, right? Their population number will decrease. Okay, eventually their population number will decrease. Whereas the grayish mouse or the black mouse here, its fur color is black or grayish, you can say, it will be able to camouflage itself. Okay, it will be able to camouflage itself against the background because of its fur color. Okay, so what is the advantage of camouflage? Can anybody tell me what is the advantage of camouflage? Okay, anybody? What is the advantage of an organism? Okay, somebody has mentioned remain unseen by predators. Okay, yes. Remember, organisms' ability to camouflage is a type of adaptation as well, right? It's sort of like a defense mechanism as well as you can say, okay, yes, somebody has mentioned increases the chances of survival. Okay, yes, it increases the chances of survival, yes, because organisms are able to hide away from predators. Okay, ability to adapt to the surrounding. Okay. Okay, somebody has mentioned protects itself from predators. Yes, right. That is the major advantage of being able to camouflage itself. That means you as the organism is able to blend in with the environment. Okay. Now, camouflage is just one type of mechanism. There are many other types of mechanisms, right? Some organisms, they produce toxins or poisons. Okay. Some organisms have a very, you can say, very bright type of coloration. Okay. They are not poisonous, but they have some very bright colorations which enables them to avoid predators. Okay. They mimic poisonous organisms. Okay. So the term there is mimicry. Okay. So these are some important terms: camouflage, right? Mimicry, the ability to produce poisons or toxins. These are some very, you can say, essential adaptations of some organisms that enables them to avoid predators. Okay, enables them to avoid predators. If they avoid predators, remember, if they have the ability to avoid predators, it increases their chances of survival. Okay, increases their chances of survival. So eventually what will happen is that the brown fur-colored rats will eventually, their population will decrease, whereas the grayish or blackish mouse population will increase. Okay, that is natural selection. Here, nature has selected the organism that has the most, or you can say is the most fittest. Okay, so here the adaptation of having a, you can say, grayish or black fur color is a favorable adaptation. Okay, is a favorable adaptation. Okay, that's natural selection. And there are three types of selection that usually takes place. Okay, there is a stabilizing selection, directional selection, and disruptive selection. Stabilizing, okay, it means it is a very stable form of selection. Okay, in this stable form of selection, the median, or you can say the middle portion, is selected. So if you have these three of these different fur color mice or rat, right, it is these three that will be selected. Okay, these three that will be selected. Or nature will favor these three and it will deselect the last one and the first one. Okay, stabilizing selection. That means in the middle, right? Stable in the middle. Directional, that means it is directed. Okay, directed towards where? Towards one side of the graph, right? Directed towards one side of the graph. That means either this side will be selected, right? Or this side will be selected. Okay, in this diagram, I've only mentioned this side just for example's sake, but this side can also be selected. Either one of them will be selected. Okay, so nature favors these two if directional selection takes place. Disruptive selection is where the middle portion is not selected. All right, the middle portion is not selected. The other two sides, the other two extremes are selected. Okay, so the organisms that fall in the extreme side, nature will favor them and they will survive. Okay, and their genes, or you can say their traits, will be carried on to the next population. Some very important terms, I've realized that some students sort of mix up these terms. Right, so some very important terms for you to understand. Species. Okay, now I've provided a very simple definition for species, right? A group of living organisms that usually have the same features, right? And are capable of breeding with each other. So you have a group of minor birds, they form a species because each of those minor birds has the capability to breed with each other. And the offspring they produce will be able to reproduce as well. Speciation comes from the word species, right? Speciation here means the formation of new species, okay? Due to some changes in environmental factors, due to changes imposed by humans, right? And some changes, like I said, in the environment, speciation may occur, where new species will come about, okay? New species will come about and they can also give rise to new species, right? And the evolutionary branch will continue to develop. Then there are two types of speciation that I want you to focus on. The first one is the allopatric speciation. Second one is the sympatric speciation. Remember, this type, the first one, the allopatric speciation, deals with geographical barrier or a physical barrier. So you have, let's say, a group of minor birds, right? You have 50 minor birds. Due to some issues, right, environmental issues, right, these 50 minor birds are separated, right, into 25, 25. They're separated now by a river or by a lake. After some time, right, this allopatric speciation does not occur overnight, right? It happens over years and years. Okay, sometimes it takes more than 300 to 400 years for it to occur. allopatric will eventually cause speciation. Okay, it will eventually cause speciation, new species to form. So that separation of that minor birds into 25 by a lake, by a mountain, by a river, or by human-made structures such as cities, roads, for example. These two populations now, they form two individual populations. They have been separated from each other for such a long time, right? Remember, the key words here is such a long time that they develop features which make them different from one another. Okay, that make them different from one another. Okay, even though they have come from the same population, they have been separated from each other due to a physical barrier, okay, or a geographical barrier for such a long time, right, that they have developed individual features or very specific features which makes them separate, therefore giving rise to speciation. Now, the second form of speciation, the sympatric speciation, there is no physical barrier. Okay, the population, that 50 population of minor birds is not separated, but speciation will occur within that 50 individual birds. Okay, so sympatric speciation, one example is polyploidy. Okay, polyploidy usually occurs in plants. It's very rare in animals, usually occurs in plants. Most of the fruits you eat, right, are an example of polyploidy. For example, bananas, right? The species that we have today are an example of a polyploidy. Okay, they're an example of polyploidy. Okay, it's where they have extra set of chromosomes, right? The cells are not able to separate chromosomes, and therefore the offsprings will have chromosomes, have extra set of chromosomes, right? They will have extra set of chromosomes. Okay, they will have extra set of chromosomes. Okay, therefore the offsprings these polyploidy individuals produce, they will also have extra set of chromosomes. Okay, sometimes the polyploidy individuals cannot produce, right? They cannot reproduce. Okay, so there are other means of them reproducing, human-induced ways of them reproducing. Okay, some important terms. So were you all able to understand those terms, or was something difficult for you to understand? If there's something difficult, you can let me know now. I will recap or I will explain that term again. Were those terms clear to you, right? Were you able to understand those words, right? Speciation is a very important term, right? Speciation is a very critical or a very important term. Okay, were you all able to understand those terms or those terminologies? Okay, remember there were two types, right? The sympatric speciation and the allopatric speciation. One deals with physical barriers, right? Whereas the other does not deal with physical barriers. Okay, is there any other form? Okay, somebody has posted a question. Is there any form of advantage the two have over one another? Okay, there is no major advantage, right? But these are, for example, the types of speciation that we observe in the environment. Okay, these are the types of speciations that we observe, that we see in the environment. Okay, there is no one, you can say the other does not have advantage over the other. Okay, one deals with physical barriers, the other does not deal with physical barriers. Okay, but polyploidy, for example, for humans itself, it has given rise to some of the important fruits, for example, that we have in our environment, right? For example, I've mentioned a couple of them, right, during the presentation. But you can say that there is no specific or major advantage that one has over the other. Okay, I hope I was able to answer your question. Okay, Neha. Okay, you can keep on typing your questions. Okay, so we'll quickly move on to the next. Okay, the types of reproductive isolation. Okay, there are many different types of reproductive isolation. The advantage of reproductive isolation is to prevent different species from breeding with each other. But you must remember, before I go down into these specifics, you must remember that reproductive isolation does not always come into effect. We do have examples of many organisms, many different species that have been able to reproduce with each other, despite having reproductive isolation in place. Okay, but nevertheless, right, this is effective most of the time. Okay, so pre-zygotic barriers, okay, pre-zygotic, okay, pre means before fertilization. Then you have post-zygotic, which means after fertilization. Okay, there's pre-zygotic and there's post-zygotic. So pre-zygotic means before fertilization. Okay, so there's temporal, there's habitat, there's behavioral, there's mechanical, and there's gametic isolation. Temporal simply means the breeding behavior or mating usually occurs different times of the day or different times of the year. Habitat is probably the most important one, right? Habitat is probably the most important one, where the organisms are separated, right? They are found in different habitats. Okay, they are found in different continents, for example. They are found in different continents. Okay, behavioral isolation, right? The behavior of organisms is different. For example, different birds have different mating calls, right? Different birds have different mating calls, which is not recognized by other species. Okay, mechanical isolation is where the reproductive structures are different and they are not compatible. So the organism may come into contact with each other, but they will not be able to copulate with each other. In gametic isolation, right, they may engage in a reproductive behavior, right? They may engage in reproductive behavior, but the gametes will not unite, right? The gametes will not unite. Okay, then post-zygotic barriers. Post-zygotic barriers actually means that fertilization has occurred, right? Fertilization has occurred. The two species have come into contact with each other, right? That is post-zygotic. But now the hybrid or the organism or the offspring, right, has come into the picture. So hybrid inviability is where hybrid zygotes, right? I think all of you know what zygotes mean, right? Zygotes are where the sperm and the egg unite, right? And they form a zygote. Okay, they form a zygote. So the zygotes fail to develop or to reach sexual maturity. Okay, and it basically dies out. Then, sorry about that. Then hybrid sterility, the organism or the offspring is born, right? And the offspring is healthy, but the offspring is not able to reproduce. It is sterile. Okay, sterile means the ability of an organism to reproduce, you can say, right? They will not be able to produce gametes that are viable. Then hybrid breakdown, right? The offspring is given birth to, right? But the offspring is very weak, right? And after some time, what will happen is that the offspring will die out, right? The offspring will die out, right? Eventually, the offspring will not reach sexual maturity. Okay? Another type that I want you to remember is adaptive radiation, right? This is where one organism or one species diversifies so much that it gives rise to an entire lineage of organisms, right? If you refer to your year 13 textbook, right, and if you refer to any biology textbook, and if you come across this particular term, right, you will see that one example will always be present, right? And this is one example that you need to master. This is of the Galapagos finches, right? This is of the Galapagos finches. So this is a group of birds, as you can see in the picture, that have come about from a single species of birds. But you can see it has diversified so much, right, that the beak shape is different from one another. These are all result of speciation, right? Eventually, they have come about through the process of speciation, right? This is a result of speciation. Specifically, this is adaptive radiation. Okay, one bird actually moved from South America to the Galapagos Islands, right? Galapagos Islands is formed of many different islands, and each island, right, you can go and do some research via Google, each island has a different food source. Okay, in one island you have an abundance of fruit trees, so the beak shape of these finches will be adapted to actually eat or consume those fruits. On the other island, the insect population is very high. So you will have birds that have a very sharp beak that is able to pierce through and consume those insects and so forth, right? Till now, we have about 13 different species of finches available, right? And they continue to diversify themselves, right? In the coming years, we will most probably see a wide variety of finches, right? More than 13. The next very important topic is evolution, right? Evolution, you can say, is a very broad topic. Okay, natural selection, the topic that we saw, natural selection, speciation, the various types of speciation. Remember, they eventually cause the formation, or they eventually cause evolution. Okay, evolution takes millions and thousands and even sometimes billions of years to occur, right? So if you look at the evolutionary timeline, right, you can always look at it up on Google, right? And you can see that organisms have moved from a single-celled microorganism to a very complex organism, right? So that is the evolutionary timeline, right? There is a change, a gradual increase, right, in complexity that also results in, that is also organic evolution. Okay, that is also organic evolution. Okay, so organisms move from very simple to very complex, right? And evolution, remember, eventually gives rise to new species, right? It gives rise to new lineages. Okay, now based on the organic evolution, okay, organic evolution concept, right? There was an experiment done so many years ago by Stanley Miller, right? A very famous scientist. What he did was that he was able to prove that organic matter or organic material was formed from inorganic material. Okay, you must remember that primitive Earth, right? Earth before the rise of all these organisms was a very unstable one. There was a lot of unfavorable gases that were present in the atmosphere, right? A lot of unfavorable gases, but there was no oxygen present, right? Oxygen came later on. Okay, so Stanley Miller was able to prove that the gases present on primitive Earth actually gave rise to the first organic material. That organic material combined together to eventually form the first cell, the first primitive cell. And then that cell became much more complex over time, eventually giving rise to multicellular organisms, such as your cell. Okay, so the Stanley Miller experiment is divided into 3 sections. Okay, is divided into 3 sections. This is the first section. You can always note this down. Number 1, right? This is number 2, right? And this is number 3. Okay, number 1 represents the ocean, right? The atmosphere or the environment, remember, on primitive Earth was extremely hot, right? So the ocean was continuously boiling, right? And you know, when water boils, it releases water vapor. Okay, so this is what Stanley Miller represented in part 1, right? He represented the ocean, right? The ocean when it was continuously boiling. So water vapor rises. Remember that this is a closed system. Nothing else is having an impact on it, right? A closed system is where no external factors are having an impact. So water vapor rises. It goes through this tubing, right? It continuously moves through and enters into this upside down flask-like structure here. That water vapor comes into contact with the primitive Earth gases that were present, right? So I've written down these 4 gases that are here: CH4, NH3, and H2, right? And H2O. These are some of the primitive forms of gases or primitive gases that were present, right, on primitive Earth. So these gases alone did not just combine randomly on their own, remember. They needed an energy source, right? Energy is needed, remember, for a reaction to take place. So the question here, okay, the question here that I'll ask you is that you must have gone over the Stanley Miller's experiment, right? So what were the two energy sources that were present on primitive Earth? Okay, what were the two major energy sources that were present on primitive Earth? Okay, you must have come across this concept. Anybody? There were two. Okay, yes, solar. Specifically, remember here, whenever you are talking about the Stanley Miller's experiment, you will mention UV radiation. Okay, you can mention solar. Okay, that's fine. But you can also mention UV radiation. Okay, there were two, right? UV radiation and lightning on primitive Earth. There were two different energy sources. Okay, so somebody has mentioned, right? Discharge of lightning. So yes, right? Lightning and UV radiation. So what Stanley Miller did was that he was able to replicate the lightning component. He used electrodes to create sparks, right? And those sparks eventually acted like lightning, and that provided the energy for the reactions to take place. So these gases have now reacted, right? And they were present in the atmosphere. So after a certain period, the atmosphere and Earth cooled, right? Representing the third phase, which is the cooling process, right? Eventually that water vapor condensed, right? And fell as rain, right? And then that actually fell down into the ocean and combined with the various other elements that were present in the ocean, giving rise to the organic matter, the primitive organic matter, which eventually gave rise to the first cell. So part 1, boiling ocean. Part 2, atmosphere. Part 3 actually represents the rain. Okay, represents rainfall. Okay. So another thing that I would like to mention, right, is the evidence for evolution. Okay, evidence for evolution. I think I've forgotten to mention it here on the slide, but I think by now you know there are different types of evolution, right? Different types of evolution that we have present, right? And that allows us to understand how evolution has taken place. Okay, and these evidences are present all over, right? You yourself are a living evidence for evolution. Okay, you are a living evidence for evolution. One example is morphological evidence, the homologous structures. Okay, this is especially present in mammalian organisms or mammals, right? You are a mammalian or a mammal. Okay, so if you look at our limb structure, especially the limbs, right? Limbs have multiple functions, especially for humans. Our limbs have multiple functions. Okay, if you look at the limb of a whale, let's say, it is also a mammalian, and a limb of a human, especially the hand bone structure, right? If you look at the arrangement, it is similar, or it's basically the same, but the function of these limbs is different, right? The whale uses its limb, specifically its flippers, for movement in water, whereas the human uses its hand for movement, also uses it to conduct various other activities, right? So these are homologous structures. Homologous structures is where the arrangement is the same, but the function is different. When the arrangement is the same, it basically means that these organisms, human, dog, bird, and whale, they've come from the same ancestor, but they have diversified so much that now we live in different environments and we use these limbs or these structures for different purposes. Okay, we use these structures, these limbs for different purposes. Analogous structures is the opposite, right? For example, wings of a bat and wings of a bird. The function of these limbs is the same. It's used for flight, okay? But they have come from different ancestors, okay? The butterfly, for example, has come from a different lineage, completely different lineage. Whereas birds, they have come from a completely different lineage altogether. Okay, just because they have the same type of feature, right, does not mean that they have come from the same lineage, right? Analogous structures. Okay, another evidence is fossils, right? Fossils are typically remains of an organism. Okay, remains of an organism. Not everything gets preserved, remember, but fossils are probably the best type of evidence that we have. Okay, the best type of evidence that enables us to observe organisms such as dinosaurs. Okay, then biochemistry, right? This comes down to your genetic material, your nucleic acids, for example, or the universal genetic code, right? Such as A, T, C, and G, right? Endocyne, thymine, cytosine, and guanine, right? This is a universal genetic code. That means all organisms have this genetic code present. Proteins, all organisms have proteins. All living organisms have proteins. Okay, remember, all living organisms have proteins. DNA and RNA, right? DNA, RNA are your nucleic acids. Okay, organisms can either contain RNA, which is a single-stranded nucleic acid, or DNA, which is a double-stranded nucleic acid, right? And this DNA basically controls your cellular functions. Okay, physiology, right? It deals with forelimbs, just like I've mentioned, homologous structures and analogous structures, right? If you know homologous structures, you will know analogous structures, right? It's the opposite, you can say. Or if you know analogous, you will know automatically, you will know homologous structures, right? So one major evidence, apart from fossils, remember, is physiology. Okay, that's one other evidence that we can physically see. Okay, whereas if you see the biochemistry bit, you actually have to do some experiments in order to obtain the evidence. Okay, comparative embryology. Okay, do not be alarmed by the word embryology. Embryology simply means the embryo. By looking at embryos of different organisms. Okay, so I have a diagrammatic form of how embryos actually look like. Right, say fish and humans. Okay, you have fish and humans. You will see that all of these organisms, from fish to humans, they come from the same lineage. Okay, now you may ask why or how. Because if you look at their embryos, the early embryo stage is basically the same. If I were to remove all the other pictures and remove the labelings here, you will see that you will not be able to distinguish between the early embryo stages, right? You will not be able to distinguish which one belongs to humans, which one belongs to the chicken, tortoise. You will not be able to distinguish. It's very difficult to do so. Okay, now that is one evidence that states that these organisms have come about from a single ancestor, right? A single ancestor, because their early stages of development is basically the same. As the organism develops, you will see certain features dissolve. For example, humans, you will see the tail and the gill slits, right? As humans, we do not require gills because we are a fully terrestrial organism. So the gill slits will dissolve, the tail will also dissolve. Okay, and you can see the limbs will start to develop, and eventually you will form a fully fledged fetus, or you can say a fully fledged human being. Same thing goes for fish. Fish are mostly aquatic organisms. Okay, they require a tail for locomotion or for movement. Okay, so the tail remains, but for humans we do not require a tail, so it basically gets dissolved. Okay, next very important thing is biomes. Okay, now biomes are simply these large areas present in a community or in an ecosystem that have a specific set of features. Okay, there are 5 major types of biomes: aquatic, grassland, forest, desert, tundra. Okay, and each one has a specific set of features. Okay, for example, a desert is different, let's say, from a tropical forest. Okay, a desert is different from a tropical forest. Now, how is a desert different from a tropical forest? What is one major difference between the two? Can anybody quickly mention that, right? What is the difference between a tropical forest biome and a desert biome? there's an obvious difference between the two. So what is one major difference between the two? Okay, tropical forest, Fiji has a good amount of forest remaining, right? Availability of water, okay, yes. Okay, desert, okay, desert has water present, but the amount of water present is very little because the rainfall is very low, right? The rainfall or the amount of rainfall is very low. Okay, somebody has raised their hand. Okay, yes. Okay, somebody has mentioned temperature. Okay, yes. In forests, usually the temperature is a bit cooler than deserts. Okay, deserts usually have a very high temperature. Okay, so that is one major difference. Okay, that is one major difference between the two, right? Temperature and precipitation or rainfall. So that's how you will differentiate between the biomes. Okay, so these are the different types of biomes. Remember, marine water or the sea and fresh water, right? Rivers, lakes, and ponds, right? They form their own biomes, right? Marine, there's a separate marine biome, there is a separate freshwater biome, right? Tropical forest, it has its own biome. Remember, the most biodiverse place or the area that contains the most biodiverse or the most biodiversity, rather, is the tropical rainforest. Out of all the biomes that is present in the biosphere on Earth, the tropical rainforest has the most organisms present. Okay, that's a very important thing that you need to remember, right? A question may be there as to which is the most biodiverse biome. So you can answer simply tropical rainforest, right? Tropical rainforest. Features that influence a biome temperature, okay, like some of you mentioned, deserts have very high temperature. Tropical rainforest have, you can say, a very average temperature. Okay, and you have the dioecious forests that have also a medium temperature, but some of these biomes, remember, experience winter. So they have a harsh temperature as well, where the temperatures may go below zero. Soil type, yes. Amount of light as well. Okay, if you look at the aquatic biome, right, usually the top layer of a, you can say, a pond receives most of the light. As you go down in the sea or the river, you can say, or a pond or a lake, as you go down, the amount of light actually decreases. Okay, and rainfall or precipitation. Precipitation simply means rainfall. Okay. Viruses. Okay, viruses. This is another very important thing that I find students have a difficulty in actually mastering. That is viruses. Remember, viruses are sort of in between living and non-living organisms. Okay, they are between living and non-living. Remember, viruses. Any virus there is requires a living host to survive. Okay, it requires a living host. If you remove a virus from a host and you leave it on your desk or table or on your laptop or PC that you're using, the virus will become inactive. And in some cases, it may die out. Okay, so viruses, they require a host to survive. Okay, and you know, because of a virus, we are having this particular revision session virtually. Otherwise, we would be having it face to face, right? And there's some other important points that I want you to remember, such as DNA and RNA. There can be a DNA virus or an RNA virus. Coronavirus, for example, is a RNA virus. Okay, it's a very large RNA virus. Okay, and they're only visible using electron microscopes. Okay, only electron microscopes. Okay, why are viruses important? You can mention this in the chat, right? I want you to write down, why do you think viruses are important? You may be asked this question during your final examinations, right? Why are viruses important? Okay, because we are currently in a pandemic. We're dealing with viruses, or we're dealing with a virus. So you may be asked as to why this particular virus or viruses are important. Okay, so this is a typical diagram of a virus. This is a bacteriophage. Okay, a bacteriophage is a virus that only infects bacteria. It does not infect other organisms. Okay, bacteriophage or phage. Okay, now virus life cycle, I will not go down into the specifics. If you want me to break this life cycle down and go over each step, you can mention it in the chat. Okay, and I will break the life cycle down into various segments and discuss it with you. Okay, but remember, a typical virus has two stages, right? Lytic cycle and lysogenic cycle. The lytic cycle is the most dangerous form, right, when compared to lysogenic cycle. Okay, lysogenic cycle is a dormant stage. Okay, lysogenic cycle is a dormant stage of reproduction in a virus life cycle, whereas lytic stage is an active stage. Okay. Next, very important thing that I'll go over very briefly, but if you want me to discuss this in detail, you can let me know through the chat. Plant kingdom. So in your year 13, you must have gone over 4 groups of plants. Okay, you must have gone over 4 groups of plants. Okay, these 4 groups of plants are bryophyta, right? Common representatives are your mosses. Then you have Petriodophyta. Quickly, anybody can mention which is the most common representative of Petriodophyta? Very common, you find it in Fiji as well, right? Some of it is a household plant as well. And then you have your gymnosperm and you have your angiosperm. Okay. Now, the first two that I've mentioned, Petriodophyta as well as the Bryophyta, these are your seedless plants, whereas gymnosperm, angiosperm are your seed plants. Okay. All 4 are fully or partially terrestrial. Okay. Bryophyta, requires water for reproduction, as well as Petriodophyta. Okay, nobody has mentioned, but Petriodophyta is your common ferns, right? The common household ferns that you have, they are common examples of a Petriodophyta. They require water for reproduction. Okay, usually Petriodophyta and Bryophyta, they only reproduce during a heavy rainfall. Okay, they have flagellated sperm, which requires water for movement. Okay, but gymnosperm and angiosperm, remember, they do not require water for reproduction. Okay, they don't require water for reproduction. They are fully fledged terrestrial plants. Okay, they are fully fledged terrestrial plants. Okay, they produce seeds. Okay, and they do not require water for reproduction. Okay, so I think I've taken up too much of my time. Okay, if you have questions regarding the viruses bit as well as the plant kingdom, you can let me know through the chat. And after Ms. Zafia has discussed her portion, I will discuss those components again. So if there's any questions, you can let me know through the chat option or the chat box. I'm more than happy to answer. I'm still available online. Now I'll hand it over to Ms. Zafia. Sorry, Zafia, I think I've taken up too much of the time, but you can start your discussions.
Hello, hi, good afternoon, everyone. So thank you, Damien, for covering those important topics for our year 13 students. Moving on, I will be sharing some information, some questions and notes on genetics. I know it's one of your favorite topics, genetics, right? So while I set up my screen, please do share some details as to where you're joining us from so that I have a good idea. So while waiting for me to share my screen, please do so. Share your information with school you're joining from and things like that. Okay. Okay, good. Thank you, guys. So good to see a lot of you from different parts of Fiji joining us. Thank you for joining us. So I'll just begin. And as Mr. Daniel was going back and forth between the PowerPoint slides and also the Google Meet platform, I will also be doing that because I cannot see the chat box while I am presenting my slides. So please bear with me as I shift between the two platforms, okay? So let's get started. Good afternoon to you all, Year 13 students, and it's good to have you here today. My name is Zafia. I am an assistant lecturer here at the University of Fiji. Today, I will be taking you through the strand of genetics. I know most of you find this topic challenging. It can be overwhelming, but trust me, it is the most interesting topic, I would say. Okay, so to be particular, I will be discussing slightly on genes, what genes are, nucleic acids, and a brief introduction to DNA replication. Before we begin with the actual revision class, let's go through an activity, right? So we'll have it less of a lecture and more of a discussion now. So before we kick start, let's attempt this activity together. I know most of you must be joining us in groups. You must be still in school. So you can attempt these in your book, write your answers in a book, and then we'll discuss it together. All you have to do is identify these structures. The keywords are present on the box here. So all you have to do is label these structures. Starting from the top box, 1, 2, 3, 4, 5, and 6. So I give you what? 30 seconds to fill in your answers, right? So once you're done, I'll switch to the chat box and then we will discuss it together. So you can label what is 1, 2, part 3, 4, 5, and 6. You have your keywords here on the box above. Six, right? So 1, 2, 3, 4, 5, and 6. Right? I'll just switch, like I said. Are you able? Okay. Okay. So are you ready with your answers? Let's start. Okay. So what do you think is structure number 1? What is this indicating? What is it? A cell. Very good, right? So it is a cell. What about 2? Inside a cell, you see another structure. What is that structure called? Good, right? So inside the cell, you have your nucleus. And inside the nucleus, you can see that there is structure number 3. What does structure number 3 refer to? Chromosomes, right? Okay. So after that, you have 4. What is 4? Chromosomes are done. The centromere, good. What about 5? 5. Right. So you can see it's faintly indicating the DNA inside the chromosome. Good. Right. What about 6? Good. It's the genes, right? So it indicates the genes. So you all are well versed with the structure of a cell and its contents. Good. So before we move on, can someone describe or explain to me what genes are? So structure number 6, we said these are genes. So what are genes? How do you define it? How do you explain genes to someone who is not a scientific, who does not have a scientific background? Good. Okay, good. So, all in all, when we look at our DNA, right, our DNA comprises of multiple thousands of genes, right? So, genes are basically short fragments of DNA that code for specific proteins. These proteins can be enzymes, hormones, or any type of proteins that is required for our survival, right? So when we look at genes, genes are short fragments of DNA, right? So as you can see, this is one fragment of the DNA that codes for one particular protein, right? We call that a gene, right? So there are two types of nucleic acid. When we look at nucleic acids are structures that are found inside the nucleus predominantly, right? So that's why we call it nucleic acid. So there are two types of nucleic acid. One is the DNA, which stands for deoxyribonucleic acid, and the other is the RNA, which stands for ribonucleic acid. So the difference between the DNA and RNA is that DNA never leaves the cell nucleus, right? That's one of the difference. Secondly, RNA can leave the nucleus, it can enter the cell cytoplasm, right? Another difference is that DNA is double-stranded, whereas RNA is single-stranded. And the most important difference is that DNA has a nucleotide called thymine, which in RNA is replaced by uracil, right? So when we look at the complementary base pairing, in DNA, adenine pairs with thymine, whereas in RNA, adenine pairs with uracil, right? So those are the common differences between the two types of nucleic acid, the DNA and the RNA. So going back to genes or genetics. Genetics is a strand of biology or a branch of biology that looks at how traits are passed on from one generation to another. Or we can simply call it the study of heredity, right? How certain traits can be passed on from one generation to another, right? So genes are basically the biological unit of heredity. You must have heard someone during your entire life must have said that, oh, you look like your mom, or you look like your father, or your uncle or auntie, right? Why is that so? Why do you look like more like a particular person and less like the other? It's because of the genetic information that you possess, right? So when we look at genes, genes are the biological unit of heredity, right? In simple terms, these are information that are passed on from one generation to another through the process of meiosis. Right? The definition of genes is that they are specific sequence of nucleotides that code for a particular protein. For example, the color of your eye is dictated by the genes that you have acquired from your parents. The type of hair that you have, either wavy or curly, it depends on the type of gene you have acquired from your parents, your great-grandparents, right? So all of the proteins in your body is coded by these specific sequences, which we call the gene. Now moving on to DNA replication. DNA replication is a process by which DNA makes copies of itself, right? So just like you make copies of notes, right? You try and make an exact copy through the photocopier. Similarly, DNA replication also tries to make exact copies of itself unless there is mutation. So why do you think DNA replication is important? What do you think is the importance of DNA replication? You can type up your answers in the chat box. Why do you think DNA replication is important? Reproduction, okay. Replicate and store genetic information. Okay. Repairing of tissues, okay. So more precisely, our cell needs to make more copies of the DNA so that it can divide, right? So for cell division, right? So let's share an analogy. Suppose you have one biology textbook, right? Only one biology textbook in the entire school. But there are 50 students doing biology, right? So to access that information, only one person will be able to use that book at a time. It would be better if we have more and more copies, isn't it? So more cells, when they have these information within themselves, the process of cell division and other repairing becomes more and more efficient, right? So when our cells divide, they also replicate the DNA with it, right? So DNA replication is very important when it comes to cell division, right? Our cells keep getting replaced. The old ones keep dying and keep getting replaced. So our cells divide. And for cell division to occur, DNA replication needs to take place, right? DNA is DNA is a structure that contains all the information that a living organism needs to survive, right? So if you do not have the DNA, your body will not know how to make a particular enzyme, a particular hormone, right? Digestion might not occur because your digestive juices or the hormones are not prepared. Okay, so DNA replication is very, very important. Are you clear so far? I hope I'm not going too fast. Okay, so once again, when we say genes, genes are short sequences of DNA that code for specific proteins, right? And these can be structural proteins, hormones, or enzymes, right? Genes are called unit of heredity because it can be passed on from one generation to the next through the process called meiosis. Only through sexual reproduction, then these genes can be passed on. If it's asexual reproduction, then there won't be any transfer of genes, right? So just to add further explanation, okay, so just to add further explanation, remember that during reproduction, you get half of the chromosomes from your mother and half the set of the chromosomes from your father, right? That makes a complete set of chromosomes for you, right? So that is the transfer of genetic information, the genes that you acquire from your parents. Okay, So DNA replication is a very important step, right? It's one of the most important processes that occur in all living organisms, right? All living organisms undergo DNA replication because it needs to keep making copies of the DNA specifically for cell division, right? So just like the name suggests, replication, replicating means to make copies, right? So how is DNA replicated? What is the process, right? In nature, the process is very complex, right? It's a very complex process, but um, it can be shortened and it can be explained in simpler terms, right? So the DNA replication can be summarized in three main steps. The first step of DNA replication is called the initiation, right? During the DNA replication initiation stage, the DNA helix will unwind, right? You all know from your previous knowledge that the DNA has a helix structure, right? It is double-stranded and it will form a helix structure. So the first step of DNA replication is the unwinding of the DNA helix. Second step of DNA replication is elongation. Elongation is where new nucleotides will come and attach to the template strand. Okay? So this is called the template strand, and new nucleotides will come and attach, forming a complementary strand. So this is the second step. We call it the elongation step of DNA replication. And finally, we have the termination, right? DNA replication needs to end. Once all the bases have been copied, the DNA replication will terminate or end, right? And that is the final stage of DNA replication, where at the end you'll have a new DNA segment. Okay. So now let's look at the three steps in more detail. Okay. Looking at the initiation stage. During the initiation of DNA replication, the first thing that occurs is the enzyme DNA helicase will unzip the DNA double strand, right? So just like if you have to photocopy a book, right? If you want to photocopy a particular page, you can't photocopy the page without opening the book, can you? No, right? So the first step, initiation, is where the DNA helix will open up, right? Allowing room for these proteins, the DNA replication proteins, to come in and start the replication process. Okay, so once again, the protein or the enzyme known as helicase will unwind the DNA by breaking the hydrogen bonds that pair up the complementary bases, right? So the ATCG bases that are bound together by the hydrogen bonds will be broken with the help of the enzyme called helicase, right? As the helicase moves along the DNA, it will continue to break more and more hydrogen bonds, therefore opening up the strand for the replication to occur. So you will have something of this sort, right? So while the DNA is getting unwound, right, while the helix is getting unzipped, on the other side, what happens is that it gets supercoiled, right? So as you pull through the strand, you can see that on the other side, the strands become supercoiled. So what happens is that it builds up a lot of tension and it can easily break the DNA, and you don't want that. So to release the tension on the other side of the DNA, you have specific enzymes again called the DNA gyrase or the topoisomerase, right? So these DNA gyrase enzymes, it releases the tension as the helicase opens up the DNA, right? So you can see that the yellow structure, that is the helicase, enzyme helicase, which is unzipping the DNA structure, while on the other side, on the other ends, you can see that the topoisomerase or the DNA gyrase is releasing the tension. Along with these two, you have these circular structures binding to the strand of DNA that has been unzipped, right? So what these do is it prevents the two unzipped DNA strands from binding together. Remember that they have complementary bases and they will try to pair up, right? To prevent these unzipped DNA from forming the helix again and to allow efficient DNA replication to occur, another structure called the single-stranded binding proteins bind to these strands so that the DNA do not get zipped together. Okay, so let's start from the top again. I know there's a lot going on in the initiation, but all it shows is how the DNA opens up. Excuse me. Right? So moving on. Like I said, if you want to photocopy a book, it needs to be open first, right? For you to access the page and photocopy it. Excuse me. So now, during the initiation of DNA replication, what happens is that the DNA helix needs to unwind, right? And that is done with the help of the enzyme called DNA helicase, right? So the enzyme DNA helicase will break the hydrogen bonds, allowing the double-stranded DNA to unzip. While the DNA is unzipping, DNA gyrase will release the tension on the other side of the replication fork so that the DNA does not break due to tension. And then the single-stranded binding proteins will come and attach, let me just pause for a while.
Uh, Thank you, Zafia. Just a reminder that the recording of these classes will be available to you tomorrow. Okay, you'll have to actually go and click on the same website that you access the link from, sorry, and you have to fill out a form actually, and you have to fill out your details, and the recording will be then available to you. Okay, so some of you had questions, I think, from my section, the virus life cycle and so forth. Okay, we'll see how much time we have left. Actually, we have exceeded the time. Okay, so I'll hand it over to Zafir. Thank you.
Okay. So sorry, please bear with me. Um, it's just the weather, I guess. So I said I stopped at the single-stranded binding proteins. So once the DNA helix is open, like the two strands have been separated, we need to ensure that they do not bind or form the helix again before the replication ends. So the single-stranded binding protein comes and attaches to this unwound helix. Okay. So after that, after the helicase has separated the two strands, another enzyme called the primase comes and attaches. Okay. Sorry, just checking the mic. So once the helicase has now opened up the DNA, then the enzyme DNA primase will come and attach to this single-stranded DNA template, right? And then this is where the replication begins, right? So this is where the replication or the addition of nucleotides will begin. So the enzyme primase has a special function because it produces a short strand of RNA. This short strand of RNA is called the primer, right? Right? So once again, once the helicase has separated the helix structure, what happens is that the primase enzyme will come and attach, right? So the red structure here indicates the primase, right? So the yellow structure indicates the helicase. So, so as the helicase is opening up the strand, you can see that the enzyme primase is coming to attach, right? And that's where the replication or elongation will begin. So before we move on, let's have some quiz exercise, right? Question number 1. A nucleotide consists of which of the following? Is it A, phosphate and a base, B, phosphate and a sugar,
D.
C, base and amino acid? Oh, that was quick. D, good, very good. So it has all the three structures, which include the phosphate, the sugar, and the nitrogenous base. Right? Question number 2, which of the following separates DNA into single strand? So we just saw how the double strand is separated. A, B, C, or D? Polymerase, gyrase, helicase, or ligase? C. Very good, right? So the enzyme helicase will open up the double-stranded DNA. Very good. So you are following through well. Moving on. Right, so now let's look at elongation. That is how nucleotides come and attach, right? So after the primer comes and attaches to this template, DNA polymerase will wrap around the template strand and it will start adding nucleotides at the 3' end, right? As you can see in the animation, right? In the animation, you can see that RNA primer is now attached, right? There is your RNA primer. Once the RNA primer attaches, then only the DNA polymerase enzyme will come and attach, adding new nucleotides as it moves along, right? So the DNA polymerase enzyme is responsible for synthesizing DNA. And this enzyme, DNA polymerase, synthesizes DNA in a continuous manner in the leading strand, right? So this is called the leading strand, right? So how does DNA polymerase add bases? Once the primer is attached, the DNA polymerase enzyme will come attached to the template strand, and then it will use the complementary base pairing system. The complementary base pairing system, which dictates that the cytosine will pair with guanine and the thymine will pair up with adenine. Right? Sorry about that. Right? So using the complementary base pairing system, which is the ATCG uh, method, the DNA polymerase enzyme will start adding bases, right? So it will keep adding bases at the 3' end, right? So you can see that it will move in a single direction only. DNA polymerase will only add new nucleotides at the 3' end, right? The arrow indicates the direction of movement, right? So this is the direction in which the DNA polymerase will move. It will keep adding new nucleotides continuously at the 3' end, right? Since DNA polymerase enzyme can only add nucleotides at the 3' end, it synthesizes the lagging strand in fragments, right? So in the lagging strand, you can see that the lagging strand will always be anti-parallel. The leading strand is from 3' to 5'. The lagging strand is from 5' to 3', right? So they are anti-parallel. And DNA polymerase can only add new nucleotides at the 3' end. So it has to go in an anti-parallel direction for the lagging strand. And that's why the nucleotides are added in fragments, or DNA synthesis occurs in fragment at the lagging strand. These fragments are called Okazaki fragments, right? Okazaki fragments because it's just named after the scientist that discovered it. So the leading strand will have DNA synthesis in a continuous manner, whereas a lagging strand will have DNA synthesis in a fragmented manner, right? So this is an animation that just shows how the primer or the DNA polymerase works, right? So on the top, you can see that is the leading strand. And at the bottom, there is a lagging strand, right? The leading strand and the lagging strand. In the leading strand, the DNA polymerase will continuously add nucleotides, right? It will synthesize DNAs, the new DNA strand, in a continuous manner. However, in a lagging strand, since the lagging strand is antiparallel and DNA polymerase can only add nucleotides at the 3' end, the DNA synthesis in the lagging strand will be in fragments. These fragments are called Okazaki fragments. Okay, so that's your helicase. The primers will come and attach, and then the polymerase will add the nucleotides. Are you clear so far? Do you want me to repeat those steps again? Are you able to follow through? Okay. If anyone is lost, then do let me know. I can cover it again. Okay, let's start again. Okay, I think this is a good diagram to discuss with, right? So when we look at the DNA replication steps, DNA replication can be divided into three main steps. The first step is of DNA replication is initiation, right? The second step of DNA replication is elongation, good. And the third step of DNA replication is termination, good, right? So looking at the initiation step. During the initiation step, what happens is that the double helix will unzip or unwind, you can say, right? So during the initiation, the double helix, the DNA structure which has a double helix shape, will unzip, it will unwind, right? So it will free the two strands so that new nucleotides can come and attach. Clear? So how does it actually unwind, right? So when we look at the process of unwinding, the unwinding of DNA is occurring through the enzyme called helicase, right? Suppose that you have a pencil case and you want to unzip it, right? Without this support structure, you won't be able to unzip it, right? So similarly for the DNA helix to unwind, it needs enzymes, right? So during the initiation, the enzyme helicase will attach to the DNA and it will start breaking the hydrogen bonds of the complementary base pairs. Once the hydrogen bonds are broken, sorry about that. Once the hydrogen bonds are broken, it will um, separate the two DNA strands. So as the strands are unzipping, it will create tension on the other side of the DNA helix. This tension is released by the enzyme called DNA gyrase, right? So DNA helix and DNA gyrase, two important enzymes, right? And to ensure that these double helix do not stick together or do not get zipped into a helix shape again, a single-stranded binding protein will come and attach to the strands, right? So unzipping of DNA through the helicase enzyme, release of the tension on the other ends through the DNA gyrase, and the use of single-stranded proteins to ensure that the separated DNA strands do not zip back together, right? So once that is happening, there is another enzyme called the primase, right? So this primase uh, forms the primer, right? The primer which helps start the elongation process, right? Okay, let's imagine you're sitting in a car, right? You're sitting in a car, you have the driver there. You have the driver, you have fuel, everything is okay, but the car is not moving because you don't have the key, right? You're sitting in a car, the driver is there, the fuel is there, but the car is not moving because you don't have the fuel in it, right? You don't have the key, right? So similarly, once the DNA has been separated, once the strands are separated, it needs to have the DNA primer where the DNA polymerase can come and attach the nucleotides to, right? So if the DNA helix has unzipped without the primer, elongation will not occur, right? Elongation does not occur without the primer because DNA polymerase needs a template as well, right? It needs a primer. to start the elongation, right? So just imagine this as a car without a key, without if there is no primer, right? So once the DNA helix has separated, the strands have separated, right? Then a primer will come and attach. The enzyme primase will form the primer, which is a short RNA sequence. It will come and attach. And then only the DNA polymerase will come and add nucleotides. Okay, so is the primer bit confusing you or is it clear now? The DNA primers?
Clear, madam.
Okay. Okay, right, so let's start from the top again. The more I reiterate, maybe it will be better for you to understand, right? So in order for the DNA replication to occur, the first step is for the DNA helix to unwind or unzip. How does it unzip? It needs DNA helicase. This enzyme DNA helicase will break the hydrogen bonds, freeing up the two strands, right? Once the two strands separate, we have the single-stranded binding proteins that stabilizes and prevents the two strands from zipping back or forming the helix again, right? And then we have the DNA gyrase, which prevents the DNA from supercoiling or it releases the tension as the DNA unwinds. Simple. After the unwinding, we have the primase enzyme, which forms a short RNA sequence, right? So it forms a short RNA sequence called the primer. The primer is where the DNA polymerase will come and attach to. And then it will start adding nucleotides in a continuous manner using the complementary base pairing system, right? So it will start adding nucleotides one by one, right? A, T, C, G, so on and so forth, right? So this animation clearly shows that process. So the green is the primase, which produces the primer, the RNA primer, a short RNA sequence. After the primer has been formed, then only the DNA polymerase will attach and start adding nucleotides, right? The DNA polymerase will add nucleotides one by one, right? So since DNA polymerase can only add primer in the 3' end, sorry, nucleotide in the 3' end, and the lagging strand runs anti-parallel, the DNA synthesis in the lagging strand occurs in a discontinuous manner, or it synthesizes DNA in small fragments. These small fragments are called Okazaki fragments. Right? So you can see the direction of arrows. They are opposite, right? Because the strands, they run anti-parallel to each other, right? So DNA synthesis in leading strand occurs continuously where single nucleotide keeps getting attached at the 3' end. However, in the lagging strand, since the lagging strand runs antiparallel, the DNA polymerase synthesizes the DNA in the lagging strand in short fragments called the Okazaki fragment, right? So helicase will unwind the DNA by breaking the hydrogen bonds. After the DNA helix has unwinded, the single-stranded protein will come and attach, right? Prevent these strands from forming the helix again. Well, on the other side, the topoisomerase will release the tension. Then comes the primase. The primase is the enzyme that will form short RNA fragments. Short RNA fragments called the primer. Once the primer attaches, then only the DNA polymerase will start adding the nucleotides one by one. However, DNA synthesis in the leading strand occurs continuously in uh, by adding nucleotides at the 3' end, while in the lagging strand, the DNA polymerase will synthesize DNA in a discontinuous manner, right? So DNA synthesis occurs in fragments at the lagging end. These fragments are called the Okazaki fragments, right? So once the DNA synthesis has occurred, once the elongation has finished, then lastly the enzyme DNA ligase will bind or glue all these fragments together, right? And in the termination step, the DNA synthesis will finish and you'll have a newly synthesized strand of DNA using the semi-conservative manner, right? So semi-conservative basically means that the the DNA strand that you'll have will contain one strand which is from the parent and one strand that will be new, right? So semi-conservative. Half of it is new and half of it is from the old or the parental strand. Okay, so moving on to our quiz. What is the function of the enzyme called DNA polymerase? Does it A, glue together the Okazaki fragment, B, add complementary nucleotides, or it unzips the two strands of DNA?
B.
Okay, good. So the answer is B. It will add nucleotides, right? So at which end does the DNA polymerase add the nucleotides to, class? What side does the DNA polymerase add nucleotides? Which side of the DNA? Okay, leading strand. Which side? So the DNA polymerase will add nucleotides both in the leading strand and the lagging strand, but it will be in different ways, right? The DNA polymerase will always add nucleotides at the 3' end, right? Always at the 3' end. However, it will add the nucleotides continuously in the leading strand and discontinuously or in fragments at the lagging end, right? So in the leading strand, you'll have continuous elongation, whereas in lagging stage, you'll have fragmented DNA synthesis, right? But in both the strands, the bases will only be added at the 3' end by the DNA polymerase. Okay, so yes, all of you have said B, it adds complementary nucleotides. Good, right? So the DNA polymerase adds nucleotides. Okay, question number 4. The fragments formed during discontinuous replication are called A, uracil fragments, B, Okazaki fragments, C,
Okazaki
primers.
fragment.
Okazaki fragments, right? So whenever you hear discontinuous, just remember that discontinuous refers to the lagging strand, okay? the lagging strand. Right? So the lagging strand will have discontinuous. There is a lag, right? There is some disruption. That's why it is discontinuous. And that is because it runs anti-parallel, right? So the lagging strand will have discontinuous replication, and that discontinuous replication is called the Okazaki fragment. Okay, so let's look at the summary together. Right, so let's look at the summary together. This is the DNA structure, which is in helix shape, right? Double stranded. Always, the DNA is always in double strand, right? So since they are in double strand, they are in helix shape. Reading the frame is very, very difficult. It needs to be open. So the first step, initiation, is where the enzyme helicase will open up the DNA, right? It will open up the DNA. At the same time, DNA gyrase will release the tension at the other side of the DNA helix structure so that it does not supercoil, so that the DNA does not break. Single-stranded binding proteins will come and attach to the separated DNA strand so that it does not zip back together. And then comes your favorite primase, right? So the enzyme primase will synthesize short RNA sequences called the RNA primers. RNA primers are very important because that is where the DNA polymerase will come and start the elongation at, right? So once the primer has been added to the strand, right? Once the primer has been added, then DNA polymerase will start adding the nucleotides at the 3' end, right? So in the leading strand, the DNA synthesis will be continuous, right? So in this case, the leading strand is at the bottom. So the DNA polymerase will add nucleotides continuously in the leading strand. However, since the lagging strand runs antiparallel, the DNA polymerase will synthesize DNA in fragments at the lagging end, right? Since they are antiparallel. So at the lagging ends, the DNA synthesis occurs in short fragments, and these fragments are called Okazaki fragments. Right? Once the DNA has been synthesized, once the elongation completes, what happens is that the gaps between the DNA fragments are sealed with the help of the enzyme called DNA ligase. Okay, so that summarizes the steps of DNA replication in a very short and simple way. Like I said, it is a very complex process, right? You all need to try and relate how these processes occur, right? As we're sitting, our DNA must be replicating as well, right? So try and relate how these processes occur in our own body, right? This will help you remember the process in a very easy way, right? So these are my references. I really did not want to overwhelm you all with too much information because I know that genetics itself is a very tricky subject for most of you, right? So please do have a read through these references. And if you have any questions, then feel free to contact us. We'll be more than happy to help you, right? So don't think that you're asking a silly question or you're asking a question that might be too basic, right? we accept all sorts of query, right? Sometimes students are not able to discuss well with us. So please, I encourage you to form groups while revising, right? I hope this tutorial session was informative for you all. I just thought to share a meme with you all since it's a common language for teenagers nowadays. Right, so if you have any questions, feel free to contact me through my email, zafian@unifiji.ac.fj, or you can contact Mr. Danian through his email as well. Right? Yeah. So I hope this session was very helpful for you all. I'll post the email in the chat shortly. Okay, all good so far. Is there anything that you want me to cover again? I know we are well beyond our time. So I'm going to share my email address with you. So please do contact us. We'd like to hear from you. I know this is a very crucial time for you because you're preparing for your external examination, right? So please do feel free to contact us. Okay, sure. So we can communicate further through email if that is possible with you, Nia.
Yeah, we have exceeded, I think, way beyond
Right.
that 1 hour
Um,
because we cannot cover everything in that 1 hour. I think most of you must have realized we can just cover the topics so much, basically. So we have gone over that 1 hour to 1.5 hour. But you can email us and we'll be more than happy to answer any of your queries or questions that you have regarding the topics we've covered or any area that you find difficulty in. And we'll be looking forward to your email if you have any questions, issues. And from our side, from the science department, all the very best for your exams starting next week. Please do your 100%. And we hope to see you this semester at UniFiji, you can say.
yeah, definitely. It was good to see you all out in numbers from different parts of Fiji. I've seen that most of you are from Vitilevu, but it was good to see most of you joining us, taking out your time. Um, so please do make use of whatever time you have available and reach out to us. We'll be more than happy to guide you and assist you with however we can. Okay.
Okay, thank you so much, guys. Take care, stay safe, and we'll catch you guys later.
Bye bye.
Bye.
Thank you, madam.
Bye.